Good morning, friends. Welcome back to biology classes. Today we'll start the remaining concept of botany, plastids. Yesterday we discussed mitochondrium, one more double membrane bound organelle. Today it is the time to discuss one more double membrane bound organelle of the eukaryotic cell called as plastids. Plastids, the meaning of plastids is in Greek term plastos equal to molds. In Greek, the plastos meaning is molds. So they are the embedded molds, and these plastids are present in plant cells, animal cells lacking the plastids. Okay, plastids are present in the plant cells, rarely in eukaryotes and also in diatoms. But all the photosynthesizing plant cells are living with the plastids. Okay, the term plastid was coined by plastid was coined by Ernst Haeckel. Ernst Haeckel in the year 1866. Ernst Haeckel coined the term plastids. Plastids are usually meant for photosynthesis in higher plants. The main function of plastids is photosynthesis in higher plants, but here they are distributed in plants for different functions also. Okay, and these plastids are full of various pigments. That full of various pigments. Okay, and based on the pigmentation, based on their colors, we classified the plastids into three types. And these plastids are usually formed by one more plastid called as proplastid. Proplastid, it is a immature plastid. Immature plastids present in this cell divides and to form a different types of plastids. So all plastids are formed by proplastids, immature plastids. So these plastids divides and differentiate to form a different kinds of plastids. Based on their coloration or based on their pigmentation or presence or absence of pigments, we classified the pigment plastids into three types. Based on their pigments or color, we classified the plastids into three types. Okay, those are chloroplast, chloroplasts. These have chlorophyll pigments. Chlorophyll A and B are abundant. Along with that, some carotenoids. In carotenoids, you find xanthophylls and carotenes. So, because of these chlorophylls, these are the green colored plastids. These are the green colored plastids. These are green colored because of presence of chlorophyll A and chlorophyll B. There are different types of chlorophylls: chlorophyll A, chlorophyll B, then chlorophyll C, chlorophyll D, chlorophyll E also. But in higher plants, we found chlorophyll A and B as well as carotenoids in the chloroplasts. Okay, the green in color. Then chromoplast. Chromoplasts. Chromo means colored. These are the colored plastids. These are the colored plastids. Chloro means green, chromo means color. They are the colored plastids, and these are of different color. They may be orange, red, or yellow colored based on the pigmentation. And pigments present in these chromoplasts are xanthophylls, carotene, xanthophylls, carotenes, phycobilins. Phycobilins. In phycobilins, we have two types: phycocyanin, phycoerythrin. Okay, phycobilins. So these gives characteristic red or yellow or orange color to the parts of the flower. You know the carrot. Carrot is orange in color, brown in color because of the carotene pigments. You know the flowers. Flowers are of different color to attract the pollinating agents. The coloration because of the chromoplasts. You know the orange fruit. You know the apple. You know the banana. Different colors are there on the fruit. It is because of the chromoplast. And these chromoplasts are usually formed by the chromoplast. Chromoplasts lose their chlorophyll pigments, and remaining pigments will be carotenoids. So because of presence of only carotenoids, the green colored fruits or flowers are what are called as leaves also turns to yellow or orange or red colored because of the lack of chlorophyll pigments. So these chromoplasts are usually formed by the chloroplasts. Okay, they present in the which part of the plant? Flowers. Flowers, fruits. Flowers are also different colored. Fruits are also different colored because of the chromoplasts. Then last one, leucoplasts. Leucoplasts. Okay, leucoplast. Leuco means white. Okay, you know the leukemia. Okay, so leuco means white. Here, these are the colorless plastids. No pigments present in these plastids. No chlorophyll pigments, no carotenoids, and no phycobilins. So these are the colorless plastids. Being colorless plastids, these leucoplasts store different kinds of organic molecules. Different kinds of organic molecules. So based on the organic molecules stored into this leucoplast, we find three different terms. Three different terms. Those are leucoplasts. Now classified into three different terms based on their stored food. Here, first one is amyloplasts. Amyloplasts. These store the starch. Store what? Starch. Whatever the food stored into the potato tuber is nothing but amyloplast. Okay, the food stored into the potato tuber is in the form of amyloplast. One more plastid of leucoplast is eleoplasts. Okay, these eleoplasts store what? Store lipids, means oils and fats. Store the oils and fats. So the seeds of sunflower, seeds of groundnut, seeds of sunflower, seeds of groundnut store the oils, and that is in the form of eleoplast. One more plastid is eleroplast. Eleroplast store the food in the form of proteins. Store the food in the form of proteins. maybe in the form of crystals or in the form of granules of proteins. These are found in the endosperms, endosperms of maize, wheat, rice, etc. Endosperm is one of the nutritious layer, nutritious tissue found in the seeds of maize, wheat, rice, etc. Because of presence of endosperm, these seeds are called as endospermous seeds. So, an endosperm is full of aileron layer, and that layer is full of protein. Okay, chloroplast is a protein. So, there are three types of leucoplast. Very, very important concept in NEET as well as they asked so many times this question. Explain three types of leucoplast based on their food stored. Okay. Also, they asked about all these three types: chloroplast, chromoplast, and leucoplast based on their pigmentation and based on their colors. So remember, they are very, very important concepts. Now, we'll discuss the ultrastructure of the chloroplast. Okay, because out of all these three plastids, maybe a chloroplast or a chromoplast or leucoplast, chloroplasts are very, very important because they only synthesize food in the form of carbohydrates. The all organisms in nature are depends on those chloroplasts because they only synthesize the food. They store the food in the producers. Producers are given by the consumers and all those. So all organisms on our earth are depends on the chloroplast because these are the sites of protein synthesis. These are the sites of synthesis of food whatever required for the all organisms. So let's discuss the ultrastructure of chloroplasts. Ultrastructure of chloroplast. Here, chloroplasts are green colored plastids present abundantly in higher plants as well as euglenoids as well as rarely in diatoms. Okay, and these chloroplasts are covered by two membranes, means there are also double membrane about organelles. And as mitochondria, these also called as semi-autonomous cell organelles. Mitochondria also semi-autonomous cell organelle as well as the chloroplasts are also semi-autonomous cell organelles. Okay, we'll study their morphology. Before that, you know the chloro means green. Okay, and plastos means molds. So these are the green colored plastids. Occurrence present in the all plants, euglena as well as diatoms. Okay, their number. The number of chloroplasts varies from species to species. It ranging from 1 to several. Okay, this number is only 1 in chlamydomonas. Chlamydomonas. Okay, and it is 1 to 16 in some species of Spirogyra. From 20 to 40 in mesophyll cells of leaves. Mesophylls of leaves. Upper epidermis, lower epidermis, in between one more tissue is there. That tissue is called as mesophyll cells. That is full of mesophyll cells. These are the photosynthetic parenchyma cells. Mesophylls are what? Photosynthetic parenchyma cells. So these cells having about 20 to 40 chloroplasts. Okay. Then their shape of the chloroplast. Okay. Usually in higher plants they are this shaped. In higher plants they are commonly they are this shaped. Sometimes cylindrical or oval. Very rarely they are cylindrical or oval. But in algae, we find different shapes in the chloroplast. Okay, in algae, we find different shapes of chloroplast. They are cup shaped in case of Chlamydomonas. They may be lens shaped. They may be ribbon shaped. They may be irregular. They may be network-like, means reticulate. Like that, we find different shapes of chloroplast in the various species of algae. Okay, they are cup-shaped in case of Chlamydomonas, lens-shaped or ribbon-shaped in case of Spirogyra, irregular in case of Eudogonium. Like that, we find different shapes in the algae, but in higher plants we find common chloroplasts are disc shaped or rarely they are cylindrical or oval. Then discuss size of the chloroplast. The size ranging from 2 to 4 microns or micrometers in their breadth and 5 to 10 micrometers in their length. Okay, the size of the chloroplast also variable, 2 to 4 microns in breadth or width or 5 to 10 microns in length. So it is the morphology of the chloroplast. Okay, they are present in all plants. Leaves of the all plants are green portion of the plants. Also present in euglena, also present in diatoms. The numbers ranging from 1 to several, only 1 in case of Chlamydomonas, 1 to 16 in case of Spirogyra, and 20 to 40 in case of mesophyll cells of leaves. Mesophyll cells are the photosynthetic parenchyma cells present at the mesophylla region or below the upper epidermis and above the lower epidermis. Shape, they are disc shaped in commonly in all higher plants. They may be cylindrical or oval, but in algae we find different shaped chloroplasts. They may be cup shaped, lens shaped, ribbon shaped, or irregular or network-like. Means reticulate. Size varying from 2 to 4 microns in breadth or width and 5 to 10 microns in length. Okay, now discuss all the parts of the chloroplast. Other structure of chloroplast is very important. So this is the ultrastructure of chloroplast. Here, as we discussed earlier, chloroplast also double membrane bound organelle. There are two membranes. The membranes are divided as outer membrane and inner membrane. Each one is about 60 to 90 angstrom in their width. Okay, and as we discussed in mitochondria, the outer membrane of chloroplast also more porous and inner membrane is less porous because outer membrane is full of porins. Porins are the protein lined vessels, protein lined carriers, and those are present at the outer membrane of mitochondria. So that is more porous and allows several molecules to enter the chloroplast, and inner membrane is less porous. So both these help the entire chloroplast. They give a particular shape and size to the chloroplast. Then coming to the actual region of the chloroplast is stroma region. Region is stroma, also called as matrix. This stroma is a site of dark reaction of photosynthesis. Dark reaction of photosynthesis. It is full of several enzymes required for the dark reaction of photosynthesis. Dark reaction, also called as our Calvin cycle, we will learn in next topic called as photosynthesis. Okay, and also have enzymes of protein synthesis. Protein synthesis. Enzymes of protein synthesis, enzymes of dark reaction are also present in the stroma region. Apart from this, stroma also have large number of starch granules, and other raw materials required for protein synthesis. The miscellaneous things present or invisible things present in the stroma region. This is the site of dark reaction. Dark reaction is glucose producing reaction that occurs in the photosynthesis, and it has several enzymes which are not visible in labels, which are required for a dark reaction and required for the protein synthesis. But from this, it having starch granules, number of starch granules, and also have large number of raw materials required for the protein synthesis as well as glucose synthesis. Then apart from this, it having a DNA, stroma having DNA called as chloroplastidial DNA called as CPT DNA. CPT DNA. It is also circular in nature, circular and naked. Circular and naked, because the presence of this DNA as it involved into replication as well as transcription, the chloroplasts are also called as semi-autonomous cell organelles. Okay. Apart from this DNA, it having RNA, several RNA molecules, maybe mRNA, discussed yesterday in the chloroplast also, rRNA and tRNA. Different types of RNA also present in the chloroplast, stroma of the chloroplast. Apart from this, we have ribosomes. Ribosomes are of 70s ribosome in mitochondria. In chloroplast as well as in prokaryotes, we find 70s ribosomes. Okay. Then here we find one of the structural unit of the chloroplast called as thylakoids. We are finding green colored structures. Green colored or plate like disc like structures in the stroma region are called as thylakoids. Thylakoids are the structural and functional unit of the chloroplast. Okay, these have capacity to synthesize different kinds of pigments and have capacity to trap the solar energy. Okay, and these convert the solar energy into chemical energy. So they are called as thylakoids. They are called what? Thylakoids. We find one more structure in the stroma called as thylakoids. Thylakoids. These are the structural and functional unit of the chloroplasts. Okay, and these thylakoids are disc shaped and have a single membrane. They are also covered by single membrane. Membrane within a membrane only present in the chloroplast. Means membranes of thylakoids are present in the membranes of chloroplast. Okay, they are surrounded by single membrane, and these are arranged one above the other like a stacks of coins, and they form a structure called as granum. A group of thylakoids arranged one above the other like stacks of coins is called as, they form a structure called as granum. The plural form is grana. They form a grana. So these grana are the actual site of light reaction of photosynthesis. You learned stroma is the site of a dark reaction. So these grana or granum are the sites of light reaction because the granum having number of thylakoids. The number of thylakoids ranging from 10 to 100 in each granum, and these thylakoids have a capacity to trap the solar energy as they having different kinds of pigments in their wall region, in their membranous region. Okay, and these grana or these each granum is connected by or joined by one more membrane-like structure called as stroma lamellae. Called what? Stroma lamellae. Stroma lamellae also having pigments, also having different photosystems on stroma lamellae, and these connect to gram. The region is called as stroma lamellae. Okay, stroma lamellae. So these many structures are found in the ultrastructure of chloroplast. Two membranes, outer membrane and inner membrane. Okay, outer membrane is more porous as compared to inner membrane because of presence of porins. P-O-R-I-N-S, porins, as present in mitochondria. Then we find the matrix of chloroplast called as stroma region. So stroma is full of different kinds of enzymes required for photosynthesis, required for direct reaction of photosynthesis, required for protein synthesis. Apart from that, having raw materials required for all those processes. Apart from that, the matrix have its own DNA, a circular naked DNA called as CPT DNA, meant for chloroplastidian DNA. It is chloroplastidian DNA, and this DNA is able to undergo replication, undergo transcription to synthesize various RNA molecules, and also involving protein synthesis. Because of presence of this DNA, the chloroplasts are self-controlled for several processes. But the partially nucleus only control the shape and size or morphology of this chloroplast, but most of the activities of this chloroplast are controlled by the DNA called as CPT DNA. Then coming to the some structures called as ribosomes. Ribosomes involved into the protein synthesis. All three types of ribosomes are found: mRNA, RNA, and tRNA. Okay, then we find, ribosomes are 70s ribosomes here, and RNA found are three types of RNAs: mRNA, RNA, and tRNA. Ribosomes are 70s ribosomes, as present in mitochondria we find 70s ribosomes. Then main structures of this chloroplast is thylakoid. Okay, thylakoids are single membrana bound structures, are structural and functional unit of the chloroplast, and these arranged like a stacks of coins as one above the other and form the one more structure called as gran. The plural form is gran. So these grana are the site of light reaction. Light reaction is a light harvesting reaction. These trap the solar energy and convert into different producing powers like ATP and NADPH. We will discuss in photosynthesis topic. Then we find the connecting links between the what you call as grana, two grana, that is called as stroma lamellii. It is also having some pigments and also involved into light reaction. Okay, so this is the ultrastructure of chloroplast. Now it is time to discuss functions of chloroplasts. Function. The main function is photosynthesis. Photosynthesis is the process of conversion of H2O and CO2 into a glucose molecule in the presence of sunlight. Okay, it's the photosynthesis is the main function of chloroplasts. Okay, then consumption of CO2 is one more function. Is the raw material of this photosynthesis, and one more function is release of oxygen. They release the oxygen. Okay. Then this perform one more function called as formation of a synthesis of starch. Okay, their main function is formation of starch by the process of photosynthesis. But main function is photosynthesis. In this, they consume the carbon dioxide, release the oxygen, as well as they give us a starch. Okay, so this is the main function of chloroplast, or everything about the chloroplast. Remember the plastids, three types of plastids: chloroplast, chromoplast, and leucoplast. In leucoplast, again we have three types: amyloplast, eleoplast, eleroplast. They store different kinds of food. Now discuss the ultrastructure of the chloroplast. They will discuss the diagram. Very, very important concept. A 5 marks question in theory, and several neat questions will be asked in this concept. Next organelles are ribosomes. As we discussed earlier in previous classes, ribosomes are non-membranable cell organelles. They are not having any membranes. They are present in the form of particles. They are present in the form of granules. Okay, these were discovered by George Palade in the year 1953. In the year 1953, George Palade observed and discovered and coined the term ribosomes. Okay, ribosomes are made up of ribonucleic acid. They are made up of ribonucleic acid means RNA and several protein molecules. Their size ranging from 15 to 20 micrometers. 15 to 20 microns. Okay, 15 to 20, sorry, nanometers. Ranging from 15 to 20 nanometers. Okay, and ribosomes are the protein factories. They are called what? Protein factories. Ribosomes are called what? Protein factories. They evolve into synthesis of proteins. Okay, they join with the mRNA, join with the tRNA, also having a ribosomal RNA, and they evolve into synthesis of proteins. Hence, they are called as protein factories. Protein factories. Okay, and ribosomes are of two types. There are two types in ribosomes. There are two types in ribosomes. ATS ribosome. One more is 70S ribosome. Common, there are two types of ribosomes, ATS and 70S. Here, each ribosome is made up of two subunits. They are made up of two subunits, and here ATS ribosome having two subunits. It is larger subunit called as 60S, this one 60S, and smaller subunit called as 40S. See here most of the students want to plus them, want to add them. It is not the 40 plus 60 and equal to, it becomes 100. Okay, don't plus them. You are going to observe their density. You are going to observe their sedimentation coefficient. You are going to observe the sedimentation coefficient. Smaller subunit is 40S and the main ribosome is of 70S. Okay, and this S is meant for Swedenborg coefficient, also called as sedimentation coefficient, and these subunits are measured in the form of their density as well as their size, and they are fixing to form only one ribosome, that form is a ATS ribosome. Okay, and these ATS ribosomes are found in the cells of eukaryotes. Cells of eukaryotes having this ATS ribosome. Cells of eukaryotes having the ATS ribosome. Okay, cells means cytoplasm as well as on the surface of endoplasmic reticulum, rough endoplasmic reticulum having this ATS ribosomes. Okay, then coming to 70S ribosome. 70S ribosome have two subunits. Those units are 30S, smaller one is a 30S, and larger one is 50S. 50S. Okay, here this is a 50S and this one is 30S. Okay, both together form a 70S, not 80. Okay, don't plus them. You are going to observe only their sedimentation. They are sedimented. They are settled in a Swedenborg's test tube. Okay, based on their sedimentation, based on their density, based on their size, they named them as a 50S and 30S. But both are the subunits of 70S ribosome. So these 70S ribosomes are found in all prokaryotes, and also found in mitochondria, also found in chloroplasts. These are the organelles of eukaryotes, but also having 70S ribosome. Having the 70S, this ATS, this is 70S. So these 70S ribosomes are found in the all prokaryotic cells like bacteria and all those. Mitochondria, chloroplast. Remember, ATS ribosomes are found in the cells of eukaryotes, cytoplasm or nucleoplasm, or on the surface of endoplasmic reticulum. Rough endoplasmic reticulum is so named because of the presence of this ATS ribosomes. So ribosomes, the main function of ribosome is to synthesize proteins. Hence, they are called as protein factories. Ribosomes are called as protein factories as they evolve into synthesis of several proteins. Okay, this is about ribosome. They are observed by George Pallet in the 1953, and these are made up of two components. Ribonucleic acid means RNA, and several about more than 40 to 50 protein molecules will be there in the structure of these ribosomes. Then the size about 15 to 20 nanometers, and there are two types of ribosomes, ATS and 70S. S stands for Swedenborg's coefficient or sedimentation coefficient. There are two subunits in the ATS ribosome named as larger subunit 60S, smaller subunit 40S. And in 70S ribosome we find two subunits. 50S is a larger subunit and 30S is a smaller subunit. They are found in eukaryotes as well as prokaryotes. These ATS ribosomes are found in the eukaryotes, and all prokaryotes, mitochondria, chloroplast having 70S ribosomes. Okay, next we have one more organelle or component of the eukaryotic cell is cytoskeleton. 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 Cytoplasmic network. One more meaning is a network of cytoplasm. Okay, in cytoplasm you learned different organelles are present. nucleus is present. You may know all these organelles must and should orient to different regions for their particular function. They need to orient, they need to rotate. Okay, they need to lie at their particular position. Okay, all these works are done by our cytoskeleton. So cytoskeleton is a network of microtubules, microbodies, and intermediate tubules. These three components, these three structures form a network in the cytoplasm. In the cytoplasm, that is called as cytoskeleton. If you draw one cell, you are going to draw this nucleus at center in animal cell. So here you find mitochondria, here you find other organelles, Golgi complex and all those. These are held at that particular position because of the cytoskeleton. It's a network in the entire cytoplasm. So these are the organelles, and cells have particular shape because of the cytoskeleton. Discuss the functions of cytoskeleton. This gives mechanical support, very, very important. Mechanical support to this cell, involving motility of organelles. Motility. Okay. Involving orientation and distribution of organelles. And one more last function is what they give particular size and shape to the cell in which they are present. So the main functions of cytoskeleton. They are made up of three components: microtubules, microbodies, and intermediate tubules. And their function is to provide a mechanical support, to provide a motility to the organelles, and also involve into orientation and distribution of organelles. And lastly, they involve into they give a particular size and shape to the cell. Okay, thank you. Tomorrow we'll discuss remaining concepts.